I, Nora Wendt, researcher at High-Resolution Instruments, am presenting to you our findings of the preprint Oxygen Dependence of Photosynthesis and Light-Enhanced Dark Respiration studied by High-Resolution PhotoRespirometry. With the next-gen O2K, the next generation of the core technology of High-Resolution Instruments, a new feature has been included, the photobiology module. The following processes and parameters can be measured using the O2K: photosynthesis, dark respiration, light-enhanced respiration, oxygen dependence, oxygen kinetics, hydrogen peroxide production, mitochondrial membrane potential, ATP production, pH values, and calcium and nitric oxide. Measurements can be performed using living cells, permeabilized cells, tissue homogenate, isolated mitochondria, and isolated chloroplasts. The experiments were run using algal cultures of Chlamydomonas reinhardtii, wild-type strain WT12. The liquid cultures were autotrophically grown in tris medium at 25 degrees Celsius and at 100 micromole per second per square meter. A dilution series of 3, 6, and 9 million cells per milliliter was created. Each dilution group was loaded into both chambers of 1 O2K per experimental run. Results: oxygen flow is a function of light regime and oxygen concentration. The net oxygen production rates are the red and the green traces corresponding to the left ordinate, whereas the right y-axis shows the oxygen concentration to which the blue traces correspond. The oxygen production rate was stimulated from dark respiration at normoxia to a maximum by stepwise increments of light intensity from 10 to 350 micromolar per second per square meter. Light-enhanced dark respiration was detected as a negative maximum of respiration after a period of light. The first trace shows the evolution of oxygen concentration while the O2K chambers remained closed. Oxygen concentration increased to hyperoxic levels due to photosynthetic activity. In the second trace, oxygen concentration was prevented from reaching severe hyperoxia inside of the O2K by intermittently opening the chambers. The same sample of the equally concentrated cell culture reached different levels of maximum oxygen production at the same light intensity. Depending on the predominant oxygen concentration inside each reaction chamber, this indicated that photoinhibition was directly linked to oxygen concentration. Oxygen dependence of net photosynthesis and light-enhanced dark respiration. Oxygen fluxes were expressed relative to dark respiration measured in the beginning of the experiment. This normalization yields flux control ratios entirely independent of cell counts. Independent of dilution, net photosynthesis was inhibited gradually from normoxia to severe hyperoxia by up to 40%. No differences could be identified comparing measurements taken in the morning or in the afternoon or between the different dilution groups. Rates of light-enhanced dark respiration measured at normoxia and hyperoxia were 3- to 4-fold higher than the initial rate of dark respiration. Light-enhanced dark respiration did not significantly depend on oxygen concentration, opposed to net photosynthesis. Summary: photoinhibition has been identified as an inevitable side effect of photosynthesis. Our results indicated that photoinhibition was directly linked to high oxygen concentration. With increasing oxygen concentration, photoinhibition became more pronounced. A significant correlation of light-enhanced dark respiration and oxygen concentration was not indicated. Thus, if light-enhanced dark respiration was an equivalent to light-enhanced photorespiration, then its inhibitory effect of net photosynthesis would not be significantly oxygen-dependent either. Therefore, the impact of photorespiration thus is smaller than of oxygen-dependent photoinhibition in our results. Further, Cell shading was a less relevant cause of photoinhibition than high oxygen, as no differences between the dilution groups could be identified in terms of oxygen production rates after normalization. Thus, further investigations of light-enhanced dark respiration and photoinhibition have high potential for efficient algal exploitation. Thank you for your time.